Okay, so let's continue. Now, another major and important thing in free radical formation is reperfusion of the organ. Now, when an organ becomes ischemic, just to a degree that the cells are still viable, they are still reversibly injured, and the blood flows back into that organ again, it could actually become more harmful. Now, there are a couple of mechanisms of why reperfusion or ischemia reperfusion injury happens, and the first being that when we cut the blood supply on an organ, we cause some type of mitochondrial damage, and mitochondrial damage actually leads to partial reduction of that oxygen, and so it leads to the formation of superoxide and hydrogen peroxide and hydroxyl and so on. It leads to the formation of reactive oxygen species. So they will accumulate more than the cell can handle to clear, and they will cause cellular injury. Now, another important thing is another important mechanism of why that happens is that the period of ischemia can cause a defect within the clearance system of that free radical. So basically, it will also accumulate and cause cellular damage. Now, another important thing is toxins. Now, toxins, these are particularly high yield. Now, toxins or chemicals, when they go through the body, for example, let's take some chemical that is really high yield, and this is called carbon tetrachloride. Okay, now this was used in the cleaning industry, but now it is banned because of that harmful effect. Now you see, when carbon tetrachloride goes into the blood, okay, it goes into the liver, and we know that when it goes into the liver, there's something that is called a cytochrome P450 within the liver, and within that system, within the P450 system, it gets converted into something that is called a CCL3, a carbon trichloride. Okay, and this is a free radical that is really damaging the carbon trichloride to the liver. Now you see, in the initial stages of carbon trichloride being in the liver as a free radical, it will cause reversible damage to the cell, and the hepatocyte, will actually swell. Okay, and that cellular swelling actually within 30 minutes or to, all the effects take place within 30 minutes to 2 hours or so, and so we will get a swelling within the rough endoplasmic reticulum that has the ribosomes. Okay, and so the ribosomes, if this rough endoplasmic reticulum swell, basically what happens is that the ribosomes will pop off, leading to a decreased protein synthesis within the liver. Now, the liver physiologically accumulates fat and redistributes it to all tissues. And so, when it accumulates fat, when the fat must go out, it must be bound to something that is called an apolipoprotein. Okay? And when protein synthesis goes down, this means that the apolipoproteins go down, and so the liver will actually accumulate fat, and the fat can't go out into the body. And so this will cause something that is called a fatty liver. Now, I actually have a picture right here of a fatty liver. So this is a normal liver. Okay, you can see that these are the hepatocytes, the canals, the canaliculi between them, and the triads and the central veins and so on. And this is a normal histology of a liver, and this is, you can see the fat accumulation right here. Okay, so this is hepatocytes, and these are adipocytes. Okay, and this is called a fatty liver. Now, acetaminophen toxicity can cause the same thing. Okay, because an overdose of acetaminophen can result in free radical formation, and it can also result in liver damage.